I'm pleased to welcome Dr. Rina Eiden. She's a professor of psychology at Penn State and received her PhD from the University of Maryland in applied developmental psychology. She then spent 27 years, did I get that right, at the University of Buffalo before she moved to Penn State in 2019. Her research focuses on understanding developmental trajectories among children at risk for maladjustment, particularly due to adversities linked to parental substance use and early childhood interventions aimed at lessening risk. And her studies, many of which follow cohorts from birth to adolescence, which is a big feat, examine developmental mechanisms that can explain the association between the parental risk factors and child outcomes. Her work has been continuously funded by NIH for a very long time, and she is an associate editor at Child Development. And today she'll be presenting us parental substance use and developmental outcomes, implications for prevention. So please join me in welcoming Dr. Eiden.
Thanks so much, Erin. And Rick, if you listen to this later, thank you so much for inviting me. And, it's a pleasure to be here, and I've had a wonderful morning. And Megan, thank you for organizing the day. So, I wanted to share some studies I've been doing on parental substance use and, sort of these long cohort studies, which means taking a group of families with children and then following them across time, looking at developmental trajectories and outcomes, etc. And applying what we've learned from those studies to early from prevention. So I got into this field entirely by accident, sort of by accident. So as a graduate student, I was in an applied developmental program, and as a part of that program, PhD program, we had to do these 10-hour internships in different, sort of field placements to get hands-on experience with different types of families experiencing different types of adversities. And my internships were with Maureen Black at the University of Maryland in Baltimore. And I was working in a clinic, we called it the SPICE clinic, but it was to prevent child abuse and neglect among Ivy drug-using families. And it was a huge learning experience. And I was responsible for doing developmental assessments in that clinic and also administering adult attachment interviews with substance using mothers. And I came away from, I was there for about two and a half years, and I came away from that experience thinking, I went in with a sort of a preconception in my head about, what substance using moms, substance using individuals might be like, and I came away realizing that these are moms who are really survivors, who had experienced significant levels of adversity and trauma in their lives. And, at the end of the adult attachment review, you end with saying, asking parents about what their hopes and dreams are about for their children. And the answers I was getting from these moms who were living in very difficult circumstances were virtually identical to interviews that I was doing with middle-class families as part of my dissertation. And so it was an eye-opening experience, and I realized, I think it made me realize the complexities that go into sort of trying to understand what substance abuse is like, what might be true for families' lived experiences, and how it might transmit from one generation to another. So there's a lot that's been written about addiction, and I like some of these quotes because they kind of underscore the idea of substance use disorders as a remitting, relapsing kind of disorder that may have lifelong sort of implications. So, why should we care about substance use? Is really a problem. And so I thought I'd spend a couple seconds just talking about prevalence. Prevalence rates for parental substance use in families is really difficult to estimate accurately because there is this huge stigma associated with substance use disorders, right? And same for mental health. Substance use and mental health issues often co-occur. And also because a lot of these families are really nervous about having child protective services involved in their lives and having their children taken away from them. But national surveys on drug use and health estimates that 1 in 8 children cohabit with parents who have a substance use disorder. And you can see what I'm not quite sure where to stand, so I'm not obstructing you. You can see what the prevalence rates are for different types of substances with regard to prenatal substance exposure, substance use in pregnancy. The one thing I would point out is that although a lot of these prevalence rates are about a single substance, that polysubstance use of more than one substance, is fairly common, and it's especially common among heavier substance users, and especially common among illicit substance use, people who have illicit substance use disorders, for example, cocaine, methamphetamine, and opioids. Parental substance use disorders, as I mentioned, is a marker for a whole host of co-occurring risk factors, including genetic, temperamental, family, and contextual risks that together pose significant risks for developmental outcomes for children. So this is a theoretical model that we use quite a bit in our lab. And we call it the transactional cascade model, but it really incorporates a lot of different developmental frameworks, including a developmental psychopathology framework that suggests that there are multiple pathways to a particular risk condition or developmental outcome, and also the idea that not all children who are exposed to a particular risk condition will have the same negative outcomes, that there's heterogeneity in outcomes. So the variables in this model are not always variables that we actually measure, but variables constructs that we keep in mind are important as we are trying to understand the phenomena. So for instance, when we're thinking about maternal substance use in pregnancy, we know that maternal substance use in pregnancy is associated with higher risk for poor nutrition, higher levels of maternal stress, illness, infection, etc., which has bidirectional associations with maternal brain and endocrine responses, which has implications for fetal development, including fetal stress and central and peripheral nervous system issues, which taken together predict neonatal outcomes, including acoustic characteristics of neonatal cries, soothability, and ability of the neonate to maintain a balanced state. These then have implications for outcomes in infancy, including individual differences in reactivity and regulation that may be measured across multiple domains. So adrenocortical, hormonal domain, the autonomic nervous system domain, as well as behavioral domain, attachment and cognition, and then they then set the stage for success or failure or adaptation or maladaptation for the next stage of development, childhood, and development of adolescent risk behaviors. There are transactional or bidirectional associations with the caregiving context. So even though that's a little box because I couldn't fit in a bigger box, the caregiving context really makes a huge difference in children's outcomes and may act as a risk factor, sort of an additive or exacerbating risk factor, but could also be really protective. And then, of course, there are genetic variations and epigenetic adaptations that also play a role. So one of the first studies that I wanted to share with you today is called the Maternal and Child Health Study. And when we started this study around 2000, there had already been decades of, at least a decade of work in this area showing that prenatal cocaine exposure was not associated with a syndrome or with sort of huge, these marked cognitive deficits, like, for example, we knew about alcohol. And there was a growing recognition that perhaps the impact of cocaine maybe more specific to this domain of arousal and how children are able to regulate their arousal when faced with challenge. And so we really wanted to focus on that particular aspect of development and think about and understand how individual differences in children's ability to regulate arousal in challenging contexts might then lead to this cascade of developmental effects on self-regulation, social competence, leading to risky behaviors in adolescents. We wanted to focus away from, there's somebody on Zoom who wants to take it. We wanted to focus away from teratology alone, so understanding, we had decades of work using animal models, showing that there was a biological mechanism by which cocaine could affect children's development. But we didn't want to just focus on the biological vulnerability, but also on the transacting risk and protective factors in the environment.
Just a question about the term. What does the arousal and teratology mean?
Okay, so arousal is, when you're faced with challenge. For example, you have an exam coming up. Do you feel your heart rate speeding up or your hands getting clammy? That's your body. So those are all physiological responses that you're having. You could have an emotional response of having more anxiety and a physiological response, right? So that's what I mean by arousal. Teratology is a field of science that is trying to understand how a particular prenatal experience might have a biological effect on fetal development.
Thanks.
Ed, would you define it that way?
That's what we do.
Okay.
So.
So we wanted to understand also the caregiving context for these children. So this is the general sort of flow for how we recruited the participants. The main thing I want to point out here is that we did I don't know how to point it here. We did a lot of intake screens. We screened a lot of families. And we ended up, it was a case control design. So we had 119 families who were recruited into the cocaine group and about 101 families who were recruited into the control group who were demographically similar. Demographically meaning similar in income, education, et cetera. And both groups were using other substances, mainly cigarettes and alcohol. And the rates of cannabis use were pretty similar across the two groups, but the cocaine-using moms were using more alcohol and cigarettes compared to the control group moms.
Did you get the information of what the father was using?
A lot of these moms were single,
I see.
and so we did a family history, and whenever we used, the family history data to look at infant outcomes, they didn't account for unique variants when maternal variables were in the model. Okay, so this was the longitudinal waves of assessment. We assess the children every 6 months between birth and kindergarten, but these were the major waves that you see here. And then again, as they entered school, so about 3 months after they entered kindergarten, again in 2nd grade, and then in early adolescence and late adolescence. So about half the children were girls, families, this was a very diverse sample, mostly Black, and because by virtue of the 2 urban hospitals that we were recruiting from, mothers were closer to 30 years of age on average, as I said, mostly single and low income. So one of the first questions we wanted to ask was, does prenatal cocaine exposure account for unique variants in autonomic regulation in infancy? So the autonomic nervous system has 2 branches, right? The parasympathetic and the sympathetic. The sympathetic is, when you feel your heart rate speeding up, et cetera. That's an index of sympathetic response. What you see here is parasympathetic response, which is measured by respiratory sinus arrhythmia in this case, which is the beat-to-beat variability in heart rate that's associated with respiration. And what we found is an interaction effect of prenatal cocaine exposure and child sex at birth, so that among boys, you see a stronger association. So this decrease in RSA from baseline, which is they're just sitting there and watching a video, to challenge, which is a frustration situation where babies are really frustrated. And you see a decrease in RSA is adaptive in typically developing samples, which is what you see in the control group. And what you see in the cocaine exposed group is this increase in RSA in response to challenge from baseline to challenge. And this difference is more marked among boys compared to girls. And maybe this autonomic response is happening because of what we see behaviorally. So trial 1 and trial 2 are the two trials of the arm restraint. That's what's on the x-axis. So you show the baby a very attractive toy, you restrain their arms for 30 seconds, then you let them play with the toy again, and you do it over again. Presumably, the amount of stress or frustration for the baby increases from trial 1 to trial 2. And so what you see here is latency to anger. So how quickly are babies responding with anger to this frustration situation? And in the control group, which is the green line, you see that as level of frustration is increasing from trial 1 to trial 2, there is no significant change. That's a non-significant line. No change in latency to anger. So they're not quicker to respond, quicker to react negatively to this frustrating situation. But in the control group, as the level of stress is increasing here, in the cocaine group, they are quicker to respond to this frustration. And maybe they're quicker to respond to this frustration because their number of different regulatory strategies they're using is not changing from trial 1 to trial 2. So this pink bar over here is the cocaine exposed group and is non-significant. But in the control group infants, as the level of stress is increasing, they're trying to use different regulatory strategies. So looking at mom, looking away from the toy, self-soothing behaviors, etc. So they're trying a lot of different things to sort of calm themselves, right? Whereas the cocaine exposed babies were not. So another sort of aspect of regulation and reactivity and regulation that we examine is this cortisol response. Right, cortisol is a hormone that we all have, and as we experience stress, our level of that hormone goes up. Unlike the heart rate or autonomic system, which is very quick to respond to challenge, the cortisol, the HPA system we call it, but really this hormonal response is much slower, right? So about 15 to 20 minutes after you're exposed to challenge, your cortisol goes up, and then another 15 to 20 minutes after the challenge is over, it goes down. And what we see here is an interaction of prenatal cocaine exposure and child biological sex at birth on this percent change in cortisol from before challenge to after challenge. So again, the interaction is the effect is more pronounced for boys. Is pronounced for boys, but is significant for boys, but not for girls. So you see this difference between the control group boys in percent change is much smaller compared to the cocaine exposed boys. Well, that makes sense? Okay. So as children developed, I'm just showing you little snapshots of results across time. As children developed, our outcomes and predictors of outcomes became more complex interplay of this biological vulnerability and contextual processes. So here, what I'm showing you here is an interaction of the stress response that I just showed you in infancy and parenting on child behavior problems at toddler age. So among children with low levels of cortisol reactivity who didn't have a huge hormonal response, cortisol response to challenge, there was no association between cocaine exposure and child behavior problems. But among kids with toddlers with high cortisol reactivity, there was a significant indirect association between cocaine exposure and child behavior problems via parenting. So mothers who had used cocaine in pregnancy had lower levels of warmth during caregiver infant interactions and in infancy, and then the higher levels of let me rephrase. Mothers who had used cocaine in pregnancy had lower levels of warmth in infancy, and lower caregiver warmth in infancy was associated with higher levels of child behavior problems at toddler age. As children got older, we were interested in change over time and developmental trajectories over time. So these are, this is the work by Danielle Molnar, who was a postdoc in the lab at the time, is now faculty at Brock. And she was really interested in developmental trajectories of externalizing behavior problems in the sample and what explained those trajectories. And essentially, what you see in the blue line is what's normative and has been reported by a number of other studies in different samples, and it's what happens typically, right? So externalizing behavior problems are behaviors like hitting, kicking, biting, aggressive behaviors, and attention problems. And usually kids normatively will increase in those behaviors until about age 3, after which time you see a sharp decrease in those behaviors. And essentially what happened with so there was an interaction of environmental risk, which is a cumulative risk variable of child and maternal exposure to violence, maternal psychological distress, and caregiving instability. That is amount of chaos and lack of routine in the caregiving context. There was an interaction of that variable and prenatal cocaine exposure, so that among children who had no cocaine exposure and low levels of environmental risk, they followed a fairly normative trajectory of child behavior problems, externalizing behavior problems. But among children who experienced cocaine exposure and high levels of caregiving risk, that green line, they did not show that normative decline from about age 3 until early school age. My allergies are really acting up.
So is this also related to kinesis? I mean, just the outright activity of the child?
It is, so activity level is a good question. We measured activity levels separately in a different observational context and have not seen, we see associations with activity level with tobacco exposure, but not with cocaine. Okay, so children are getting older. So this is, they're in kindergarten now, and by kindergarten age, about 30% of the sample had been in foster care, alternate care, non-biological parent care at some point between birth and kindergarten, right? And so these children, we had written a paper before this showing that children who were in foster care were experiencing lower levels of maternal psychological distress or caregiver psychological distress, lower levels of violence exposure. Their contexts were a little better from a risk perspective. And these are teacher reports of child behavior problems, teacher reports of hyperactivity, attention problems, and conduct problems in the school setting. And what we saw was a significant difference between children who were in the cocaine group but had remained in biological parent care, the pink bar, and children who were in the other two groups, who were in foster care, cocaine exposed, but in foster care, and the control group infants. So just to note that the children who were cocaine exposed and in foster care were actually more heavily exposed. So they tended to go into foster care right from the hospital. They were discharged into alternate care, or they went into care a little bit later, but they were the more heavily exposed children. And so one takeaway of this result for us is that if you could improve the caregiving context, so these were also children who remain in biological care, were exposed to substances postnatally as well. So one takeaway is that if you can improve the context in which these children are being raised and provide treatment for substance use, then that might have positive effects on children's development. So children are now in middle school, right? And this is the work of Eiden, who was a postdoc in the lab, now faculty at Texas A&M. And he really wanted to test three different models, conceptual models of predicting conduct problems. Conduct problems are, again, aggression related. And so he examined a cascade model where A predicts B, predicts C, predicts B across development, or an additive model, which is A and B and C together predict D, or increased risk for D, and a bidirectional or transactional model that they all impact each other, and found support for a cascade model where essentially two parallel cascades for risk for conduct problems. One is through maternal depression. So I'm not showing you the full model here. I've taken out things. So depression and I've taken out the infancy variables for simplicity, but there was continuity of caregiver depression across time. And prenatal substance exposure was associated with higher risk for being in non-biological care, so placed in foster care. Children who were in foster care experienced lower levels of caregiver depression. Children who remained in biological care experienced higher levels of caregiver depression. And this continuity of caregiver depression from infancy to toddler years was associated with higher conduct problems at preschool age, and then there was continuity over time in conduct problems. Right, so that was the one pathway through caregiver depression, depressive symptoms. The other pathway was through this caregiver sensitivity pathway. So prenatal substance use was associated with higher levels of socioeconomic adversity in infancy, which was associated with lower caregiver sensitivity at toddler age, which then predicted preschool age caregiver sensitivity, leading to lower levels of conscience in kindergarten, to higher levels of teacher-reported conduct problems. So conscience is internalization of rules of conduct, is the way we measured it. So we measured it in the lab through a cheating game, and I had a video that I took out, but it's essentially it's a game that you ask children to play, but you set up the rules in a way that there's no way that they can win the game. But you stack the deck against them, and you tell them so there's a dartboard, and if they get the ball in the center of the dartboard, they get this really cool prize. If they get the ball on the sides, they get this okay prize. And all children cheat, right? I mean, they want the cool prize, but what we're measuring is not whether they cheated or not, but really how long it took them to cheat and how many different rule violations that they made. And so this is a marker of self-regulation, right? It's the ultimate goal of socialization. What we all want our children to do is behave in normative ways according to contextual expectations. I'm sorry. So we want them to behave in ways that follow the social rules, even when we're not watching them. And so that we think is a really important marker of self-regulation that then predicts a whole host of, here I have conduct problems, but it also predicts other related variables. So now the children are in adolescence, and remember the cortisol graph that I showed you before, where the cocaine-exposed boys had higher levels of cortisol reactivity compared to non-exposed boys. And what we see here, this is a person-centered analysis of cortisol reactivity in adolescence. We had them do a Trier social stress test, which is you have to come into this lab and give a speech task and a math task, and there are judges wearing lab coats who you have to do this in front of. It's very stressful. It would be stressful for any of us. And what we have in the x-axis is just time. So 0 is, they've just rested they come into the lab, they relax for 20 minutes, they're listening to music, watching video. And then we start the Trier about 10 minutes in, so time 2. Cortisol, remember, is a very slow moving system. It takes 15 to 20 minutes to peak. So this is about that time when it should be peaking, and then this is recovery, the last time point. And so we found 3 different profiles of cortisol reactivity. One is this elevated profile, the green line. You see the green line. And the other is sort of this moderate reactivity profile, which is majority of the kids, about 65% of the sample. And then we also had a blunted profile or non-reactive profile, which is about 19% of the sample. So there were no direct associations between prenatal cocaine or any other substance exposure and cortisol reactivity in adolescence, but substance exposure was associated with early life adversity, and there was an interaction of early life adversity and caregiver sensitivity on both the probability of being in the elevated and the blunted profile. So under conditions of high early life adversity and high maternal sensitivity, the probability of being in either one of those profiles was much lower, but under conditions of low maternal sensitivity, there was a much higher probability of being in one of those profiles. So it was an additive effect of early life adversity and low maternal sensitivity on being in one of the 2. And a lot has been written in the literature about too high or too low levels of reactivity being problematic. So our next question really is to see how these profiles then relate to later adolescent outcomes, which we haven't done yet. This is the work of Kristen Perry, who's postdoc in the lab right now. And she's really interested. These are kids living in very high risk environments, right? So they're exposed to a lot of, they're living in low income environments, a lot of neighborhood violence, et cetera. The schools are not that great. And so we wanted to look at gang involvement and weapon carrying in this sample. And those are 2 variables that Kristen's trying to predict. And really the main predictor of gang involvement was externalizing problems in early adolescence and predicting late adolescent gang involvement. And remember that externalizing behavior problems in early adolescence was really that green line of kids who were not showing that normative decline from age 3 to age 5. There was continuity or stability of externalizing problems for that group. She also found an interaction of neighborhood risk and engagement with positive peers. So this interaction surprised us because it was really positive engagement with positive peers was associated with lower likelihood. So this bar here of weapon carrying in high risk, but not in low risk neighborhoods. So this protection of who you're hanging out with really became apparent in the context of a neighborhood that you're living in, if it's a high risk neighborhood with high levels of crime and violence. This is just a note that we're really involved in these are all the students in the lab right now, and they're really trying to understand sort of the cascade from birth to adolescence. We just finished the adolescent data collection, late adolescent data collection not that long ago. So there's a group of students who are using the paper chase method. If anybody has questions about that later, ask me, to write a paper on resilience. So when children are doing better than they might be expected given their prenatal exposure and their postnatal circumstances. We measured activity space in early adolescence, and we're really trying to use that data to understand kids' neighborhoods. So they were using a web-based mapping technology. They were asked to drop pins on the different locations that they frequented during weekdays and weekend days that we then used geographic information systems to connect to crime level data. And we're trying to then incorporate that into our understanding of individual differences in children's functioning. So that's kind of interesting. Maggie, who's here, who's in the lab, is really interested in co-occurring internalizing and externalizing problems, how anxiety, depression, children who experience high levels of anxiety and depression might also experience high levels of aggressive behavior problems, and the co-development and co-occurrence of those issues might be predictive of more negative outcomes than either one alone. And so she's really trying to understand how that pattern of behavior problems predicts late adolescent outcomes. So what might be some prevention implications of all of this? So one for us is that protective processes may operate at many different levels, right? It could be at the contextual level, it could be individual difference variables like self-regulation, could be parenting, and that inner, if you think about Bronfen-Renner's circles, the inner circle. That our second sort of takeaway is that promoting positive caregiving experiences, even in the context of prenatal exposure, may be promotive and protective. We also think that addressing maternal mood symptoms and emotional dysregulation, Liz Conrad, who's now at Duke, has done a lot of work and written a lot about emotional dysregulation and how that might be a transdiagnostic factor that underlies a lot of mood disorders, but also substance use disorders. That might be a powerful sort of target of intervention. Also that we need to begin early, like that cascade of risk processes happen really early. So, this takes me to I just want to mention that I'm collaborating with Mary Dozier, and we have a clinical trial called Modified Attachment by Behavioral Catch-up Trial, which is for low-income families with opioid use disorders, and it's a downward extension of attachment by behavioral intervention that is evidence-based and has been used with a lot of families living in adverse circumstances. So I'm not going to say a whole bunch more because I do want to talk about another study, but the modification is published now, so if anyone's interested, please look them up. So I think I'm missing a slide. Yeah, I'm missing a slide, but let me just tell you about this a little bit. What time is it? Does anyone know?
Yeah.
2:40? Okay, so I need to wrap up. Okay. So the second study I wanted to briefly mention is a study of fathers with alcohol use disorders, where we started in infancy and followed the families until late adolescence. So why do we want to understand that? There's been Bob Zucker, for example, has written a lot about families with fathers with alcohol use disorders and how at the time we really didn't understand much about what happened from an environmental or family perspective with these families in early life. There's a lot of literature in adolescence. And then Bob Zucker and Hy Fitzgerald had started the Michigan longitudinal study, which began in the preschool period. But we really wanted to understand what happened in early infancy in the first 3 years of life with these families and sort of cascade beyond, and what might be some risk and protective factors that might impact children's outcomes. We knew from other literature that these were kids who were at high risk for alcohol use disorder themselves, 4 to 10 times more likely to have alcohol use disorder. And some of it might be genetic, but we wanted to understand more the environmental effects. This is just the sample of 227 families. And the times of assessment were from infancy to late adolescence.
Oops. Okay. Want to read it?
So we started when the babies were 12 months of age, and what you see over here in this column are all the different constructs that we were interested in, and we ended when it says 11th grade, but they're 11th to 12th grade of age. So I'm just gonna give you the broad sort of outcomes. What we found were direct and spillover effects from family processes to parenting. So father's alcohol problems in infancy was associated with parenting across time, but some of it was direct and some of it was indirect through higher levels of family conflict. If you have a partner with alcohol use disorder, and higher levels of verbal and physical aggression in the family. And then parenting, particularly maternal sensitivity, had cascading effects on self-regulation and externalizing behavior problems, which was one sort of pathway to heavy drinking and marijuana use in later adolescence. The other pathway was through this maternal sensitivity continuity to monitoring in early adolescence. And that related to engagement with substance using and delinquent peers, which was then a proximal predictor of heavy drinking and marijuana use. So I'm gonna skip over this one. This is just saying that we did some person-centered analyses and found different variables to either increase risk or lead to protection. And this was my slide to talk about limitations that I will skip so that I can get to this, which is what are some implications for prevention? From this particular study for us, it was that if we could combine a couple-focused parenting program with intervention for heavy drinking that reduced father's hazardous or heavy drinking, taking a harm reduction approach, we might find some positive results. We also wanted to find an intervention that focused on promoting parental warmth or sensitivity and had evidence that it promoted children's self-regulation. And we wanted to time it before age 2 because maternal sensitivity at age 2 was a particularly significant predictor of that cascade of risk processes. So this is a current clinical trial that we're involved in, that we're doing right now that I'm co-PI with Stephanie Godleski, who's a postdoc in the lab and is now faculty at RIT. And it is, what we did was take an evidence-based intervention targeted for families who are entering parenthood. So it's that during that transition to parenthood, and has evidence for promoting positive parenting, promoting children's self-regulation, and really improving family or couple relationship. It's delivered in a group format, and it's evidence-based in terms of long-term outcomes into adolescence. And so we took that family foundations intervention and thought that it was might be particularly useful because it's targeting that transition to parenting when parents might be motivated for health behavior change. It's couple-based, it's group delivered. There are 5 prenatal sessions and 4 postnatal sessions. And we were in the field for about 2 months starting recruitment when COVID hit. And so then we thought, this was like February, March, and we thought, okay, we just have to keep everybody trained until August. It'll be all over by August, and we'll be good to go. Right. So by August, it was very clear that that wasn't happening, and so we had to do this switch to virtual format. So now we're in the field. We're recruiting in Pennsylvania, Maryland, and New York State. And we're doing everything virtually, which has been a whole another experience. So this is just what the classes are about. And it was, so these are the strong foundations classes. This is the control group, the healthy foundations classes, which is psychoeducational. So parents are randomized to one of two conditions. And it's been also a challenge because these are not parents who are looking for treatment, right? These are not fathers who think they have a problem. So this is not a good intervention for families or fathers who are very heavily alcohol dependent. They need supervision, more supervised, more intensive treatment. This is for fathers who are heavy drinking or have some level of dependence but don't have a physiological dependence. And so we took combined family foundations with screening and brief intervention for alcohol use and are delivering that in using a motivational interviewing format, which is really trying to motivate people to think about health behavior change in a very non-threatening and engaging, interactive manner. None of the intervention content is lecture-y in any way. So the goal of the intervention is not to tell parents what to do, but to help them, provide a context for them to think about and talk with each other about health behavior change and talk to them about what research shows in terms of alcohol effects on the family, et cetera. And there's a lot of role playing and building and giving them practical strategies to avoid alcohol in different contexts, et cetera, et cetera. And these are some of the outcomes. So we're assessing family functioning at mid-intervention, which is about 1 month post-natal, to about 3 years. Some additional future directions that I'm happy to talk about, but I'd rather have some time for questions and discussion. And I want to end by saying none of this work is mine, really. It really takes a whole lot of people to do this work, and people who are much smarter than I am. So, and thanks to NIH for years of funding. So that's it.
Yeah, we got time for at least 10 minutes for questions.
I had a question about I'm sorry, go ahead.
Oh, sorry, I was just gonna ask, so with the study that you guys did on infant stress reactivity, I was just wondering if you had any ideas for why there was a difference between sex at birth?
Yeah, that's a really good question. It's a good question for Ed, too. So this is, our interpretation is that boys are more biologically vulnerable to a whole host of prenatal influences. And so that might be part of what we're seeing, is that prenatal exposure by sex interaction is particularly significant for boys.
I saw in your bar graphs that there was a typical sex difference in the controls.
Mm-hmm.
And so that was actually ended up being reversed.
Mm-hmm.
So, and we see that, well, I'm an animal person, I deal with rats and zebrafish, but with the rats, we do see a lot of this attenuation or even reversal of typical sex differences with exposure to pesticides, exposure to cannabis, exposure to nicotine. And so it may be a common thing about kind of desexing of the brain.
Mm-hmm. Yeah. Does that help?
I had a question about therapeutic drugs. So as these kids may show a kind of an ADHD-like syndrome in terms of increased risk-taking and attentional problems, hyperkinesis, are they kind of put into that sort of medical stream of getting prescribed stimulant drugs, and what are their reactions? Is that an adverse treatment because they have this history of developing under a stimulant drug?
Yeah, that's a very thought-provoking question. I don't know if I have the answer. A lot of these a lot of, in the cocaine sample,
Yeah.
I would say probably, I mean, even in the control group, because they're exposed to a bunch of other things as well. We did measure how many were on medication, and by second grade, I would say at least 45 or 48% of the sample were on ADHD
Is that,
meds.
that's incredible.
Mhm. Like for the sample as a whole,
Yeah,
right? And I can't remember if there was a group difference in terms of the percentage, but yes, they are. And there are also a lot of the sort of on the phone, spending time on the phone with families, time that I've personally spent with families has been to try to get them to get into behavioral treatment for ADHD. And so,
interesting.
yeah, and how much of that is truly ADHD and how much of that is reaction to context?
Yeah,
You know,
and there's a huge, gender difference. And boys are much more prone to be diagnosed with ADHD because it's a pain in the rear diagnosis. Kids that are pain in the rear get
get
diagnosed.
diagnosed with
And
ADHD.
so if
yeah,
boys are
right.
running around the classroom, then they get diagnosed. If girls are quite inattentive but sitting quietly, then they don't.
Right.
Just a question, I think it was a cocaine study, and you mentioned how those who had parents like who abused substances, who didn't go into foster care were, I think, more likely to be diagnosed with depression than those who went into foster care.
So those who didn't go into foster care, who remained with their biological parents, had higher levels of hyperactivity and inattention.
So I was just curious because, I don't know, just in the courses I've taken, like, there's been quite a, I guess, maybe some criticism of the foster care system. Was there any specifics on, like, was this like a typical foster care?
Yeah, no, that's a very good question. I mean, I keep reminding myself to say non-biological care because not all of these children were in foster care, really foster care. So this is the sample is predominantly Black, right? This is the strength of Black families. Familism is a huge strength. And so a lot of the children were actually in kinship care. And some of them went into adoptive care, right, from birth.
So
And
that
so-
counts as,
I think so, yeah, that all counted, all of them we put in that category of not remaining with their biological parent.
got it. Okay, thanks.
Good question.
There's a question in the chat. Two actually. Okay, we can start first with Elizabeth. And I'll read it aloud so other people can hear it. But in many of your models, maternal sensitivity emerged as an important mediator of child mental health outcomes. Does maternal sensitivity operate similarly in your ABC intervention for mothers with opioid use disorder?
Oh, good question, Liz. Yeah, I think so. I mean, the ABC is really focused on that is the main target or mechanism for intervention, right? So the goal is to promote nurturance, and it is an open question because part of the value of doing a randomized clinical trial is to see if the mechanism that you think is causative, is causal, really is causal, right? So we have promising data. So that Tvachnik paper was focused on autonomic regulation from the pilot sample, and really at least with targeting maternal sensitivity in particular in ABC, we see more optimal autonomic regulation among the opioid use disordered families. So yeah, I think so. Yes. So the answer to your question, Liz, is yes.
Okay, we have some more. We have one more chat question, but are there other questions in the room or?
That question. I mean, cocaine is a sympathomimetic, and it causes kind of a stress response. So these outcomes that you see in the kids, in other studies, they're just looking at psychosocial stress, during pregnancy, do you see a similar kind of array of effects in the offspring?
Yeah.
You do? Okay.
Yeah. Yeah, so we have actually not yes and no. Are you talking about prenatal stress that's
Yeah.
different
Stress
from
during
exposure, stress during pregnancy that is unrelated to substance exposure?
Yeah, right. So
Yeah.
basically, does cocaine give kind of a drug-induced stress response that is kind of similar to a psychosocial stress response?
Yeah, that's a good question. So Lori Washlag group had a paper not that long ago where they had tobacco and cannabis exposed children and prenatally stressed children, and the effects on children were very similar across both groups. And so and you know, much has been written. The translational neuroscience perspective, for example, suggests that really prenatal substance exposure is a marker of toxic stress. Like that's essentially what it's a stress model from a fetal perspective, rather than an exposure specific, like a stress as exposure specific leading to different outcomes. I don't know that we've really done that research though, you know, so ideally, to answer your question, you might want a model or study where you have the cocaine exposure, for example, group that you contrast with no substance exposure but psychosocial stress group. And I don't know that who are similar in demographics, right? So that's I think that is one of the most difficult challenges is the context is so different for some of these demographic contexts.
And if people find these ameliorative treatments, the results, adverse effects of psychosocial stress during pregnancy, maybe those would be also similar to beneficial treatments after cocaine during pregnancy.
Yeah, I do think Liz is onto something with this idea of focusing on emotional dysregulation as a transdiagnostic factor, because I think that could be sort of the bridge between all of these different exposures, including prenatal stress.
Okay, we could do this last one. Julia, I was wondering if you could talk more about the MABC project and what the key modifications were for adapting the ABC for a high-risk opioid use disorder sample.
Yeah, of course. So one of the big modifications for ABC was ABC started at about 6 months of infant age. And so our initial conversations were all with Mary Dozier, who developed the ABC, were really about what can we do to start it early in the third trimester prenatal period so that we can, it felt like it was too late, like missing the window of the first 6 months was missing a key opportune time for promoting positive change. And so one of the big changes is that the MABC begins in the third trimester of pregnancy. And so sort of preparing moms and to transition into that role, into that parenting role. And the other modification involved like MABC is every week, MABC is every 2 to 3 weeks to try to give more flexibility around. So these are all mothers who are on medication-assisted treatment for opioid use disorder. So give more flexibility around the ebbs and flows of that. They're also low income, so they're like dealing with survival issues as well as substance use disorder. So it's just a little more flexible and relaxed approach. I think it's a home visiting program just like ABC is. So I think some of the other changes had to do with the nitty-gritty of the intervention content. I think we have more on the MABC about following the child's lead and how to strategies for promoting optimal regulation in children who are experiencing a lot of physiological dysregulation. due to opioid use, opioid exposure. So.
Okay.
Thank you.